Hello students, welcome to the class. Today's topic is on do's and don'ts in the plant tissue culture laboratory. So we'll be dealing with two different things here. Things to do in tissue culture laboratory and things not to do in tissue culture laboratory. So let's look into the points. What are the things to do in plant tissue culture laboratory and move into the things not to do. So here we have in point number one we have do plan and prepare for every laboratory exercises before coming to the laboratory. That means you have to do homework. That means you have to prepare for every laboratory practical classes before you move into that, and for that you have to read into the manual, and you have to ask your technical assistant or your teacher in charge for that, and the teacher in charge will definitely give you the work plan, and according to that you will be going through the practical classes or your research. Okay, so in point number two we have always be neat and tidy yourself when you work into tissue culture lab, because contamination is a danger in plant tissue culture laboratory. You have to maintain total aseptic condition in plant tissue culture laboratory, and that's why it's important to maintain a tidy and neat and tidy clothings. I'm not saying that we have to wear new clothes every day, but we have to at least maintain clean clothes, wash clean clothes every day. Okay, so the next thing here is lab trays to be properly leveled for its effective use. Never carry tissue culture lab trays outside unless for washed. Okay, because sometimes these tissue culture trays are meant for keeping toxic waste, or sometimes it is used for waste. So the waste which is coming out from the tissue culture laboratory or any biotech laboratory is not safe for exposing or disposing into the environment. So it is better to keep it safe and dispose it in a proper way in biohazard bag, or you have to seek proper knowledge about disposing the waste. Okay, the next thing is point number four, do wear proper attire. Okay, that means you have to wear the uh, the, the prescriptions which are uh, given by the technical assistant or the lab in charge. Okay, which is according to the requirement of the laboratory. That means first of all we have safety glasses or goggles to protect from UV rays or the harmful toxic chemicals which might enter your eyes. Okay, and next thing is lab coat. That means also called as apron, and here gloves, chemical resistant or gloves. Okay, to whenever you have to handle toxic chemicals like HCl or NaOH3, okay, or any acidic or any carcinogenic agents, so you have to better wear these gloves to protect yourself from future harms. Okay, and then it is advisable to wear long pants rather than short pants. As you can see here, you are exposing here whole of your leg, and of course the shoes. Okay, wearing shoes is always recommended, leather shoes rather than cloth shoes, because when the chemical spills or if there is any chemical spill, it might harm your feet, and because it is exposed here, as you can see here. And always be professional and not to be casual. As you can see here, casual working, routine casual working may harm your skins, or it may do permanent damage on your skins. Okay, so better be prepared before before there is a permanent damage on your skin. Okay, the next thing is number five. We have to always sterilize all the items you deal in the tissue culture lab, including your coat, cap, and mask. Okay, so the thing here is use of sanitization before you enter and in the entrance of the tissue culture lab if it is highly sophisticated one. Okay, as you can see here, the food trays here is actually chemically treated with disinfectants. Okay, and as you enter you have to rub it, or you have to dip your shoes inside this disinfectant, and you have to pass through it. Okay, so this at least ensures that there is the surface microbes which may be a contaminant to the laboratory is avoided at some certain level. Even though if it is not 100%, it may reduce at certain level. So these precautions are taken, and so you have to take, okay, from your side. And the next thing is number 6: switch on the lights and ACs in the culture room at 8:30 a.m. and off by 4:30 p.m., giving 8 hours incubation at 25 degrees Celsius. This timing may change depending on the lab conditions. Okay, the timing is given so that the culture room, again, this is a culture room, is the culture room is provided with 8 hours of light at least. So it also depends on the type of plant that you are growing. If it is a long day plant, you'll be requiring long amount of daylight, okay, that means the tube light. Or if it is a short day plant, you'll be requiring less amount of light and more amount of dark lights. Okay, so according to that, you have to maintain the lights by regulating with a time switch or a light switch. As you can see here, the amount of daylight and night light can be regulated automatically. Okay, if you don't have this, you can regulate. You can come to the lab at 8:30 or 9, and you have to leave by 4:30 or 5, or you can come at 4:30 to 5 and switch off the light and switch on the light. Okay, so you have to regulate manually, or either you have to use this time switch so as to give optimum amount of time, optimum amount of light and dark periods. Okay, the next thing is always keep the apparatus and all the lab wares needed by the site before starting the practical class, okay, or the work. So as you can see here, in the laminar airflow, the researcher is keeping all his requirements prior to the start of uh, his activity. Okay, because you don't want, after sterilization of your hands, okay, you don't want to get up and go fetch the things time and again. Okay, so this is why it's required, it is important to fetch everything on the sides and keep it there, okay, for the working period and not for the entire period. Okay, because this is not a storeroom. Yes, you have to understand, most important thing is this is not a storeroom, and you have to empty it as soon as you complete the uh, practical activity. Okay, so this is another point. And next thing is keep your workbench neat and clean before leaving the laboratory. As you can see here, I was talking about keeping everything on its place before you complete the before you leave. Okay, and it is important because if you keep the same thing inside the laminar airflow hood, okay, or the working area, there will be chaos. Okay, the next day if you enter, or somebody else if enters, he will get dumbfounded, and he will find it difficulty for him to start up the practical or the activity. Okay, so if it is a multi-using laboratory, so you have to be careful and you have to take care about other users also. And the next thing is number 9: disposal of biological materials such as microbes by sterilizing and in special containers for biohazards. As you can see here, the sign here given you is called biohazard sign, and bags having these signs are meant to say that they are contained with biohazard waste, that means the biotic waste. So you have to be careful, okay, while handling this. You always wear gloves, because you may not know, it may contains carcinogenic agents or any other toxic chemical which might harm. So you don't want to welcome those, and you have to be careful and always dispose in this bag, and the college or the university will take care of the disposal waste. Okay. The next thing is, if there is any broken glasses or this or any uh, glasswares, you have to first of all inform the technical assistant or the teacher in charge. And after that, the things to be done is disposal in a proper disposal box. Okay, it will be there in every corners of nooks and corners of the laboratory. So first of all, it will be categorized into three types. The disposal can be categorized into three types. First of all, sharp containers, The biohazard signs may be there or may not be there, but always a disposal box will be there, where or the technical assistant or the laboratory in charge will direct you to dispose it in the particular bin or box. So you have to follow that. And biohazard, next thing is biohazard bags. Okay, and then third thing is glass disposal boxes. So you have to identify properly which boxes are for disposal of which kind of materials. Okay, and biohazard bags usually comes with tissue wipes, okay, tissue paper wipes, or the cotton swabs, or the gel, okay, which are usually contained with carcinogenic agents, ethidium bromide or something. Okay, and the next thing is number 11. We have to be sure where to dispose the hazardous chemical. We have talked about this. Again, coming to the next one. Okay, coming to the next one, we have always use safety glasses or the eye protective protection. If you have if you're wearing an eyeglass, it's a protective means. But then again, if you are dealing with UV rays, or if you're dealing with carcinogenic agent, it's always advisable that you wear a lab glass, okay, which looks something like this, okay, and it is protected from 3-degree angle, and it will protect you from hazardous chemicals, acids, carcinogenic agents, and at the same time from UV rays, which might harm your eyes, okay, which might do some permanent damage. Okay, I'm not saying that this laboratory activity is quite dangerous, but it may sometimes turn into dangerous if you don't handle it, if you don't act professionally. Okay, next thing is tie back your long hair to avoid contamination and hazardous fire hazards. Okay, so things which are to be done, which are with who are having long hairs. So you have at least you have to tie your hair on the back or something, so that it doesn't come on the way of you and your practical activity. Okay, and you might not want permanent damage on your hair also. And number 14, we have be very careful with Bunsen burners, okay, because we are dealing with fire, and which is most dangerous thing. And always keep the burner at a distance from the organic solvents, okay, because organic solvent contains carbon, hydrogen, and oxygen, because which are highly inflammable. Because so when you're dealing with benzene or ethanol, so you have to be very careful, because you're dealing with you have that is why always I'm always saying it time and again, you have to act professionally, and and avoid such accidents prior to what it might happen. Okay, next thing is point number 15. Wash your hands before leaving the laboratory, even if you wear gloves. Wash with disinfectants in running water before and after the work. Okay, because this is because some of the chemicals which I was talking about, ETBR, in case of molecular biology, it is carcinogenic. It may, if you don't even though if it is not visible to your naked eye, the permanent damage may not be in your naked eye, but it may be effective when it comes to 1 or 2 years. Okay, and you may also bring it on the way home. So it's best to wash properly with soaps or detergents or hand wash, and ensure that you are not carrying those harmful chemicals, at least in your home. Okay, and the next thing is you can see here the eye wash, okay, which should be there in sophisticated labs, but let's see if we have in every lab. It's necessary, as I think it is necessary in every lab, because accidents might happen, or if something happens, you might need this. And the next thing is number 16, locate eyewash stations. Okay, this is called eyewash, and fire extinguishers, and safety showers. Okay, and the next thing is number 17, be familiar with your chemicals in the laboratory and take maximum care in handling each chemical, because if you mishandle uh, chemicals. Okay, mishandling chemicals can be in certain ways. First of all, one thing is when you take out a chemical and when you need it, you use it. Okay, and when you keep it back, you keep it in a different place where it is from. Okay, so it is a mismanagement which you have to avoid, because the different user, because laboratory is not a single user when it comes to your graduation, or if you are a researcher, it is meant for multi-user. So if you're keeping it in a different place, so the other person may find it difficulty to do his practical class or the research. So that is why it is recommended to keep it in its own place and be respectful with one another. Okay, and the other thing is keep handling of chemicals. Okay, certainly when you're dealing with hazardous chemicals. Okay, you have to ask your technical assistant first of all to what kind of chemicals are there, and he will tell you the different kinds of chemicals which you have to take special care, and chemicals which you have to which you have diluted chemicals which you can uh, take care, and which you can handle from time to time. So um, these are the things you have to take care in disculture lab while dealing with chemicals. Keep chemicals back in position after use. Yeah, that's that there is the minimum things we can do in disculture lab. And in most of the labs we use lab coats, extra books. Okay, that means the lab books or the records which we usually carry, you should keep it back into a locker room. Okay, things why? Because the lab coats, or you can call it as aprons, it may contain with hazardous chemicals if you're dealing with microbes, if you're dealing with in plant pathology, if you're dealing with uh, pathologies. So it might contain in your lab coat. So you may take it if you bring it back to your home, so you may take it along with you. So it's best to keep it in the lab itself, at least in the locker room. Okay, the next thing is do clean laminate airflow fume hood with ethanol, 70% ethanol. And the next thing is wash all items inserted into the hood with 70% ethyl alcohol. And number 22, we have you must cut your nails regularly. And number 23, we have always maintain aseptic condition while working in cultures. Okay, so these all are to maintain an aseptic condition. Okay, to make it free from microbes. Okay, because you don't want any other microbes to enter except the one you're dealing with, or except the one you're going to isolate in the laminar flow fume hood. Okay, so in plant tissue culture laboratory, we're not going to deal with microbes, but rather we are going to deal with plant tissue culture. So we don't want any microbes to enter inside it. Once entered, if it is fungus or bacteria, so it may do a permanent, or your workflow may be stopped because of that. Okay, so you have to take care of it. The next thing is after completion of work, always label the culture with names, codes, date of work. If you don't do so, what will happen? The type of the date of tissues, okay, and the date of in explant that you have treated with the media is going to get expire in no time. Okay, and if you don't label the time and the type of explant, so there may be confusions in lots of the samples. Okay, so it's best to label it right after the inoculation of explant into the culture media. Okay, the next thing is tightly close all bottles and caps. You have to tightly close. Okay, because if it is not so, so the microbes in the air might enter inside it, so it may cause again contamination. Number 26, we have removed materials from the hood after work. LAF is not a storage area. Again, Okay, this is the point here which I mentioned earlier. uh, so you have to maintain clean area right after your work, okay, so that others find it easier to start his work. And next, coming here, we have things not to do in tissue culture laboratory. Okay, let's see what are these. Don't mishandle the chemicals, solutions, street lamps, UV lights, instruments, apparatus, or electricity. Okay, so this is quite important. You have to be professional and you have to start acting like a professional when you enter into any biotech lab. Okay, this is not a joke. And the next thing is, don't roam here and there in the laboratory without work or any aim. So if you have if you enter into a room, you have to listen to what the technical assistant or the lab in charge is saying, and you have to follow that. Okay, and you don't have to roam in here and there and look into the different chemicals which are prepared or stock solutions which are prepared there and mishandle those. Okay. Next thing here is, never misplace labwares and other equipments. Okay, so thing aprons which are kept here, you don't have to keep it somewhere else so that the others find it difficult. Number four, we have, never leave the culture in your laminar airflow. Okay, so it is this is the point which I've said earlier. Then number five, we have, don't eat, drink, smoke, chew gum, or apply cosmetics in the lab. Okay, never use a microwave, laboratory fridge or freezer for food. Okay, sometimes people which are not unprofessional use lab fridge, freezer for food. Okay, because they find it easier, just because they find it easier. You For those people, it is advisable to not use it, because we are dealing with carcinogenic agents. It might do permanent damage in their health. Okay, they might get cancer because of that, because most of all we are dealing with ETBR in case of microwave, and at the same time we are also dealing with keeping carcinogenic agents in the lab in the fridge. Okay, so it might get caught, the food might get caught with it, and finally the individual might get, and the friends which are feeding them. Okay, and the next thing is, never eat or drink using lab glasswares. Okay, these are these. And don't touch any chemicals with hands, as some may be corrosive. Yes. And one important thing is, while after using every equipment, you have to enter it into the logbook. Okay, and while after using also, you have to before using you have to enter, and before after using also you have to enter. Okay, because at least this ensures that you are using it, and if something happens, if there is any damage into the equipment, you have to inform to the lab technicians. Okay, so this is quite important to avoid any damage. You don't have to be scared if there is any damage. Okay, the things will be taken care by the uh, lab assistant, but just information to the lab assistant is quite necessary. Okay, because if there is any damage which can be saved, it can be done right away. And also wipe out any chemical which are spilled, or any acid which are spilled. You have to take care with the instruments or equipment which are there in the laboratory as yours itself. Okay, next point is number 8, don't leave a burner lit unless you are standing next to it, because someone else might be injured if they do not realize it is lit. Okay, because as you can see, the Bunsen burner, the light is not visible. Sometimes the light is not visible, so you have to at least make sure that the Bunsen burner is off when you are moving out. Okay, you don't have to leave it when you go out and come back after having chit chat with your friends. And at least make sure it is off when you have to move out. And point number 9, we have, never taste a chemical as it may be poisonous. Okay, so permanent damages might be there, so you have to take care of this. Number 10, we have, never use cracked or broken glasswares. If there is any if you find any one of these, you have to inform to the lab technician right away, or the lab in charge right away. And point number 11, we have don't throw materials other than sharps into the sharp materials receptacles. Okay, we have earlier learned that we have different boxes for disposal of different materials or glasswares. Okay, so sharp materials like glass, uh, broken glasses or blades, you can dispose it into the sharp glasses sharp or broken glass bin box, or don't throw it in the biohazard bag, which is meant for swabs, cotton swabs, or uh, tissue papers. Okay, so you have to make things clear which things are which boxes or which bin are for disposal of which one, which kind of equipment. So so this is why it's important to listen to your lab technician, quite important, before you start into the, or before you enter into the tissue culture lab or any biotech lab. Okay, point number 12, we have don't place any chemicals on hand. Okay, you don't have to play. Okay, this you can think, see here is mercury. Okay, so you don't have to play with this because it may it may contaminate your hand also. It may, you may consume it also. And it is not safe for you. So don't play with the chemicals. If you see any one playing, so you have to stop them also. Numbers 13, we have don't keep the reagent bottles open. Okay, because if you keep it open for a long period of time, the chemical property may degrade due to exposure. So it's always advisable to keep it in the bottle, reagent bottle. And also when you're preparing a reagent bottle, it's recommended which chemical are you going to be there. Some bottles are transparent, and some bottles are ambrose, which is dark in color. So uh, chemicals which are chemicals which are degraded by sunlight or any light can have to be kept in the ambrose bottle, which is dark color. And chemicals which are not degraded, which are undegraded by light, which can be kept in a transparent bottle. So you have to clarify which chemicals are which, and which can be kept in which kind of containers. Okay, so the next thing is never mount pipette. Okay, so these are primitive pipettes which we use in every practical classes. I think if you're if you have deal with chemistry laboratory, you can deal with this. Okay, so if you use this for any acids, it is dangerous. Of course, it's associated with dangerous, so it's not advisable. So for using um, harmful chemicals, it is always advisable to use these hand pipettes, hand micropipettes. Okay, which comes with again different levels of capacity. So according to that, you have to choose the hand pipette and use it. Again, this is much more sophisticated one which you use in I think nowadays in every laboratory. And next thing is 15, number 15, we have don't wash solvents or hazardous materials down the drain. Don't combine chemicals for disposal unless you know them they are not reactive which one another with one another. Don't bring inflammable liquids such as alcohol near the flame. Okay, so in biotech lab, you're dealing with lots of inflammables, lots of hazardous chemicals, and lots of chemicals which are carcinogenic. Okay, so right away after after doing your practical class, you don't have to spill it into the basin, wash basin, or into the any dustbin. Okay, so you have to properly keep it into the proper waste bins, and you have to make sure that it is disposed in the right in a right area. Okay, number 16, we have don't disturb the arrangement of reagents or chemicals in the shelf. Okay, so in every In laboratory, the chemicals are kept as such in alphabetical order. So if you keep it back, if you find the chemical from here and you take the chemical after using it, if you kept it back here, so the other user might find it easier, much more easier, rather than mismanagement or keeping it somewhere else. So you have to be you have to be much more responsible for others also. And number 16, we have don't spill out chemicals. Okay, spilling out chemicals may happen accidentally, or it may be intentionally, or it may be just for fun. Okay, but the thing is, don't spill out chemical here means just make sure that it is not spilled out. Okay, you have to handle it very carefully. Okay, you have to avoid accident, so that you avoid accident, and at the same time you have to be more responsible, okay, with more professional uh, knowledge. Okay, and at number 18, we have don't keep water running if there is no use. Okay, you have to save water. Okay, you have to uh, use water efficiently. And number 19, we have don't put anything of the laboratory, example pencil threads, labels, inoculation needles, pins, etc., in your mouth, ear, nose, eyes. Okay, so contamination may be there. Okay, so maybe with chemicals or microbes. So best to avoid is to take precautions. At number 20, we have don't put your fingers in your eyes, ears, mouths while working in the lab. Okay, so at least you have to keep this habit. Okay, nowadays it's a severe time for pandemic of coronavirus, so it is, I think people are much more familiar with this and more accustomed with this. People should not keep their hands in their eyes and nose if they are going out, if they are handling if they are going out in crowd places. Okay, so that this you are avoiding the virus. Okay, so like just similar to that in every laboratory, so it is advisable to uh, not smear your mouth, nose, and eyes right during your practical class. Okay, 21, number 21, don't throw solid waste materials like filter paper pieces, test tube pieces in the sink. Okay, so this is, you can see here, there is a chaotic condition here. So this is a dirty way of management of laboratory. So always you have to keep it in proper disposal. Okay, so right again, another important point is don't play with chemicals. Okay, so with this, this is the end of the class. I hope you all are understood, and you will act responsibility with the biotech lab or pharmaceutical app. And that is all. Thank you.